in the second podcast. So I am going to merge chapters 26 and 27 together because there's so little 27 we actually have to cover. So 26 is going to really focus on how we categorize life, taxonomy. It's going to focus on how we have learned and how we structure organisms and how we classify them based upon their evolutionary lineages and histories. And so phylogeny is just a fancy word for evolutionary histories, and systematics is just this idea of taking a bunch of different evidences and trying to piece together where everybody fits all together, all the species that ever have been on this like evolutionary tree of life, if you will. And so they're very closely related. It's just the idea of, are you looking at one individual species and kind of all of its history, or are you looking kind of at intertwining all of those different species and their histories with one another? And so this just shows you here the graph. We have kind of the start of life, and you can see we have the splits of bacteria and archaea. So these are going to be the early prokaryotes. Then we've got eukarya. It's eukaryota there, but they usually say eukarya. And so you've got that domain that kind of comes up here that starts to divide off, and so you get the essentially what we have as protista. And then the protists have divided off and eventually given us fungus. They've divided off, and we've gotten animalia, or metazoa essentially is what they're doing there for animals. We've gotten the other groups that have given us algae, as well as plants that have kind of broken off on their own. And so we get this huge kind of tree of life that is just gradually developed as organisms diverge. And then those organisms diverge, and so we can get, in many cases, some pretty wildly different things. So it's kind of cool. So for phylogeny, as we're doing phylogeny and systematics, we're really just trying to make sure that we understand, once again, everybody's past and how everybody's past is linked to, or how it is structured within this network of all these relationships of what evolved from what, which evolved from what, how everything's diverged. So this is very complex, trying to go back to the beginning of life and trying to sort out just how everybody's connected to everybody else, to try to figure out, what were the steps that were part of your lineage to get you to where you are today. But that's what phylogeny more specifically and systematics more broadly are trying to do. Now, as we move on here, we've also got phylogenetics, which is going to try to kind of do the tree of life, the more literal one, I guess, where you're trying to kind of map it out. And so you're putting it on paper and trying to connect everything. And so this really, if you go back far enough, has its roots where Linnaeus was the first one to come up with a more modern taxonomic system, which really is one that just tries to organize things. Now, he wasn't doing it so much evolutionarily. He was doing it through similarities physically that things had, which later on fit pretty well, and so we've kind of just melded that a little bit, massaged it just a little bit to show these evolutionary relationships, and we've continued to do that as we discover more things and as we get more evidences to kind of make it a bit more accurate. But he was the first one that started this, and he also came up with this hierarchical way of organizing it that was really cool, where he said, all right, we've got kingdoms up at the top that's really broad, and kingdoms will have one or more phyla, or phylum if it's a single one, and then each phylum is ultimately going to have one or more class. Each class is going to have one or more orders, and so it's kind of like those Russian nesting dolls where you open one up and there's multiple, there's a bunch inside, but then you open up that one and there's another one inside. You open up that one, there's another one inside. Except in this case, sometimes you can open up a nesting doll and there might be like 3 of them inside, but they still each have smaller ones inside of them. It's kind of like having a gigantic box, and you open that up and there's one or more smaller boxes in there. And you open that up and there's one or more smaller boxes in there. And that's what we're doing with this type of system here. So it allows us to show if 2 things are related but more distantly, more broadly, that would be kingdom. And it also allows us to show if things are very, very closely related, where they're essentially interchangeable. They're able to interbreed, and that would be like species, which is the most specific. And then he also came up with binomial nomenclature, which is where he looked at this saying, we didn't have domains then. We've added that above kingdom. But even without that, you had kingdom, phylum, class, order, family, genus, species. If we want to give a scientific name and I have to rattle off like 7 names to get to it, that's not fun. So he said what we're going to do is use binomial nomenclature, where we're going to use the genus and use the capital letter for it. So for humans, it'd be Homo with a capital H. And then we're going to use the species. and we're going to do that with no caps. So in this case, it would be sapiens for us. So Homo sapiens would be the genus and species that we belong to. Now from there, you can essentially look it up if you needed to and see, all right, this genus Homo, where does it belong? And so you could figure out the family, the order, the class, etc. These still exist, but we're not going to use those for everyday discussions. We're just going to use the binomial, two names, genus, species. Capitalize genus, don't capitalize species. And if they say specific epithet, that's just another fancy word, pinky up, for species, so don't freak out. And if you see them refer to a taxon or taxa, the plural, that just means a group. So technically all of these, domains, kingdoms, phyla, class, order, family, genus, species, all of these are taxa. Any group that you have, just like any box you have, you could just say it's a box. It doesn't imply the size of it, it's just saying it's a group. So don't freak out if you see that used. It's just the general description for a group, which could be any of those. All right. Now, when we try to go through and describe these relationships and make these phylogenetic trees, which map out these evolutionary relationships, what scientists have now settled on as the best way of doing this is cladistic analysis, which they'll use to make something that you'll see is called a cladogram. They used to do like these more elaborate phylogenetic trees that looked like trees, and they straight up, and they just kind of put stuff down, and it was much more subjective. So as scientists try to make this more objective, what they've done is say, all right, let's focus on where you get these either or choices, like a choose your own adventure. So you'll see here this goes forward, and then we go up or down. We've got two possibilities. And so what you'll see here is one of these possibilities will essentially be kind of normal, I guess you'd say, and the other one's going to have some new trait, or it's going to have lost an existing trait. So like in humans, we've lost what was a tail. So you can use the lack of tail because we previously had one as a new or novel trait. In this case, it's just a regression, losing it. Versus you can also do something where you've gained something like larger cranial size or being bipedal, although you could say that's losing being more of a quadruped, but whatever. So you can use those as also being one of these defining characteristics of which direction you go. So you essentially say you have this thing or you don't have this thing. And then you keep doing that for each fork. And so it makes it fairly easy for us to go through and understand why are we separating these? What happened, what new, what great thing occurred evolutionarily that allowed for these two populations to diverge. And so we'll focus on, like if you're doing it really broadly, you might see mammals, you might say, well, they developed hair. It could be developing being endothermic, regulating your body temperature. It could be something where you have, like reptiles, the amniotic shelled egg. And so amphibians lack that, reptiles have that, and so we'll do this kind of choice there. And each of these branches, just like we said taxa before, if it's on a cladogram, if it's done cladistically, we just say that's a clade. So once again, any of these kind of branches plus whatever else is with it, that's a clade. Now, when we're trying to organize life, our goal, our ultimate ideal when we're going through creating cladograms or analyzing things is to try to line things up to be monophyletic. And what that means is it's this middle guy where we have this original common ancestor and then we have everybody that we should have. All of its ancestors are paired up with it. So this common ancestor is the common ancestor of every single thing that's been produced by it. That's what we want. But sometimes we screw it up. And this is what we're trying to avoid. So paraphyletic is where we've grouped up a common ancestor with a bunch of its offspring, a bunch of its, I guess you could say, like daughter species that have evolved, but we've missed some. And so if you've missed some of them, that's going to be paraphyletic. All right? Now, polyphyletic is where you have more than one common ancestor that's represented by what you're grouping. So if we say that we're going to have this clade, all right, so we say this is our clade, what we've covered in. If we say that, we're wrong because there's three different common ancestors that are part of that clade that we're representing. And so unless we manage to go through and go back far enough to include this D guy, we're wrong. Now, if we include the D guy, if we say that D is the common ancestor of all those, then we're fine. But if I say something like E is the common ancestor of F and G, I'm wrong. because E is not the common ancestor of those. It's technically more like a sibling, if you will. And so that would be considered polyphyletic, where there's not, at the start of your divisions here, at the beginning of this, that is not the proper common ancestor. We have to go back further, essentially, in this case, to get to that. So that's polyphyletic. We have too many of these common ancestors. So monophyletic will be our goal. This is what we're going to be after as we go through and do cladistics. We want things to be like that red example. And this is constantly being readjusted as we figure out that we're wrong sometimes. We'll, like I said, massage things a bit to make sure that we get things where we need to be. Now, this is just an example. So we've got our common ancestor, and as we go along, you're going to see that there's going to be these new traits that come up. So we've got the pipefish without a brood pouch. So you can see here, the evolution of a brood pouch is the novel trait that makes us go up. So this is the one that gives us kind of a bump. And then after that, we've got pipefish with brood pouch, but now we've got a new thing where it's prehensile tails. And so now we've got prehensile tails, and so that takes us up to the next branch. And so the next branch now goes through and it gets another new trait. And so this process will continue and continue, and that's how we want a cladogram to be, and that's why scientists like it, because we can kind of point out and say this is what ultimately has happened. And plus it makes it very simple because we don't have these crazy things like it used to be sometimes, like 12 things are coming out. We can kind of do it one at a time to make it easier to structure it in our brains. It kind of shows this progression as things evolve. To get to a more modern species. I don't say better, simple things can do great, but to one that's perhaps a bit more modern and perhaps a bit more complex. So in this case, it's just going from a relatively simple pipefish-like guy to eventually get to the seahorse, all right? And you'll see too, oftentimes these will be set up with time. So this is the older things, and as we go in this case, sometimes they'll be vertical where you go up, like through time as you go up. In this case, as we go right, we go through time, and you'll see these species are all still extant or living. In some cases, you can have a cladogram where it kind of like splits or breaks off somewhere because a whole group went extinct. So maybe none of these, ancestor-like species made it, and so you might see where it kind of stops, and that's what it's trying to tell you, is that that whole group went extinct. So you're not going to be able to find them. Now, with cladograms and cladistics in general, one of the difficult things is then trying to figure out, when we look at the evidence, to make sure that we're not grouping things together that are analogous versus grouping things together that are homologous. So if you remember, homologous means that we have a common ancestor, which is awesome if we're doing cladistics, because we're trying to group things based upon their common ancestry. But these analogous guys can oftentimes look similar. As you can see here, they start out different, but they end up looking similar. And so we have to be very careful to not let those sneak in like some early scientists did, where they would group stuff together like pterosaurs, like bats, and like birds, because they'd see those wings and go, ha-ha. But there is no common winged ancestor that has this wing that's persisted on. Bats develop their wings from wingless mammals. Birds develop their wings from wingless dinosaurs, and pterosaurs develop their wings from wingless reptiles that were not dinosaurs. And you can see when you look at them that a bat wing is primarily fingers, if you will. You can see that a bird wing is primarily forearm with some fingers, whereas the pterosaur wing is actually much more like the upper arm, if you will. It's not very much hand at all. And so each of them is structurally going to be very different from the other. And so these things are analogous. We do not want to group them together. We have to make sure that they're each in their own clade, completely separate from one another. They should not in any way come together except for a common ancestor way back in the reptile lineage at some point, but a long ways back. Now, the last bit, I think, before I'll stop this one for today, is when we set up a cladogram, we normally include one outgroup, which is one that's not what we're trying to study. So if we're doing amphibians, we might throw one type of somewhat amphibian-like fish, so a similar thing, but not quite, just for comparative purposes. So we can say that the in-group, the ones we're looking at, the amphibians perhaps in this case, we can say like this is the new thing that they got that made them different from what was previously there, which in that case is likely going to be a tetrapod or bone-finned, if you will, fish that they had. Sometimes call them lobe-fin. So we're going to have the in-group versus out-group. So this will be the most of the cladogram, but this will be like that first guy for comparison. And when we're doing this, we have to focus on what are shared ancestral traits. So ones that you have because you already had this. So before amphibians, what traits did we already have in fish that were there, that are ancestral, that everybody has because the common ancestor of all amphibians had it, versus which ones are going to be our shared derived characteristics, which will be ones that some guys might have, so like A and B might have it, but C doesn't. These are ones that are the new novel traits or regressions that have occurred that separate out the various species that are amphibians. So it's not just fish then amphibians. In reality, it's you have some fish developed in amphibians, and then amphibians diverged in a variety of different ways to give us a tremendous number of amphibians. So we've got frogs and newts and salamanders and Sicilians, I think you call them. I might say that horribly wrong. But the guys that look kind of like snakes that are amphibians. So each of those guys is going to have some characteristics that have derived characters. The other types of amphibians do not have those characters, just they do. And so those are what help us distinguish amongst these guys that are within the in-group. Now we're going to practice this in class some to make sure that you guys can set this up and understand it better, but I believe that should be what we need to get to.